So what I want to do with this little mini lecture is talk to you about some of the strategies DNA viruses use for gene expression. There's just too much to cover in class, so I thought I would do half online and half in class. So the first thing I want you to remember is how we express genes. The goal is to make protein. So you've got DNA to RNA. Remember, we're going to make mRNA and then translation to protein. A couple other things to remember: that in order to express a gene, the promoter has to be recognized by RNA polymerase and transcription factors. Once that gene is transcribed to RNA, you need a 5' cap and a 3' polyA tail. These are essential components for translation, which I'll show you in a second. And the other concept I want to remind you of is this idea of alternative splicing, where you can have one RNA with multiple exons, and different exons can be joined, and you're going to produce different proteins. So alternative splicing is a way to go from one RNA to multiple proteins. And the reason I bring this all up are that some viruses use similar strategies. So what we're really talking about today is getting to mRNA and then getting it translated. And we've talked a little bit about how to get to a positive sense RNA molecule when we talked about some of the genome replication strategies. So your double-stranded viruses, you have 4 classes, class 1, 2, 6, and 7. Okay, and these are going to look a lot like normal transcription because they're starting out with double-stranded DNA. So class 1, already double-stranded DNA, you just need to get RNA polymerase on there to make mRNA. Class 2, if you remember, it goes from single-stranded DNA to double-stranded DNA, and class 7 goes from double-stranded DNA that is partially double-stranded, actually goes to RNA, which goes back to double-stranded DNA. But as far as gene expression is concerned, all of these are starting with double-stranded DNA. So it's pretty straightforward, like eukaryotic cells do. The asterisk here is for something that I had forgotten to mention with genome replication, is that to make double-stranded DNA, the cell has to be in S phase. So it has to be going through cell cycle, because that is when DNA polymerase and the other DNA replication genes, or proteins, I should say, are being expressed. So unless the DNA virus encodes its own replication genes, such as DNA polymerase and anything else it needs, a lot of DNA viruses will actually induce S phase in the cell so that the host cell is producing those essential proteins needed. Again, that goes back more with genome replication, but I wanted to mention it. All right, so for gene expression, we're going to assume the double-stranded DNA is made and that the gene expression is happening in the nucleus. And this is true for all of these different viruses except for Poxviruses, which is like smallpox, vaccinia virus, cowpox, monkeypox. These are these large DNA viruses that replicate and express their genes in the cytoplasm. So I'll show you a little information about that. Since this is all happening in the nucleus, these viruses can use the host RNA polymerase and the host splicing machinery. So it's really straightforward. The final step in gene expression, which is translation, for all viruses, DNA, RNA viruses, all viruses use the host ribosomes. So all translation happens in the cytoplasm. The thing I want you to remember about translation, and the reason I emphasize 5' cap, 3' polyA tail, is because this is how translation happens. So these are eukaryotic initiation factors. They make a cap binding complex that will then recruit the ribosome. So this is how eukaryotic translation happens. You can see that the polyA tail is important, the 5' cap is important. This is all for recruiting the ribosome. So for DNA viruses, we don't have an issue here because the mRNAs are being made from double-stranded DNA, and using the host RNA polymerase, a cap and tail will be put on. So what we're going to talk about for DNA viruses is kind of how they control their gene expression. Okay, so this is human papillomavirus, HPV. It's a double-stranded DNA virus. And some of the interesting things I want to point out is that it encodes some of its own transcription factors. So, there we are. One of the things you have to remember is, even though translation is straightforward because it's using the eukaryotic cell information, or eukaryotic cell proteins, you still have to be able to recruit RNA polymerase to the promoters of the virus. So a lot of viruses have very few promoters. So E and L stand for early and late gene expression. So there are early promoters and there are late promoters. So these transcription factors encoded by the virus can start promoting early transcription, and then as other proteins are made, they can promote transcription of the late genes. So the late genes you can see are the capsid, which makes sense. You want to express all your genes for replication and cell proliferation. This is actually controlling cell cycle here. And E6 and E7 are proteins we'll talk about later this semester. These are ones that are actually related to the cancer-causing properties of human papillomavirus. Okay. So for human papillomavirus, the reason I'm showing you this is to understand that this virus will encode some of its own transcription factors in order to get the host cell RNA polymerase onto this genome and allow expression of the viral genes. Oops. SV40 is also a class 1 double-stranded DNA virus. Sorry, it has again 2 promoters, an early and a late. It makes only a couple RNAs. The reason I want to show you over here is that they use alternative splicing and alternative start sites in order to express the genes. So very similar to some of the strategies we see in eukaryotic cells. This is also SV40, and the reason I put this one on is I wanted to explain to you that the large T antigen is a transcription regulator. And So it helps activate cell cycle to S phase so that the virus can replicate its genome. But it also promotes early gene expression by binding onto the DNA. And then later, as it gets a lot of large T antigen built up, it binds to site 3 here and blocks the early promoter, and that turns on the late genes. So the viruses, again, are encoding some of their own proteins in order to regulate gene expression. Adenovirus has actually 4 stages: immediate, early, intermediate, and late gene expression. Lots of genes, and it also uses alternative splicing and alternative start sites. Herpes simplex 1 virus, also a double-stranded virus, a double-strand DNA virus. It has 3 phases, so it has the alpha, beta, gamma phases of transcription. And again, all of these phases are important for controlling which viral genes are expressed. You don't want to overwhelm the cell, use up all the resources unnecessarily. So if you're first expressing your proteins for replication and expressing then your proteins for capsids, you're using the resources in the cell wisely. HSV has an interesting structure in that it has this area called the tegument. Okay? And it's this area here between the capsid and the envelope, and it's full of proteins. There's about 20 proteins, they're still trying to characterize all of them, that are packaged in this in this virus. And so it packages proteins such as VP16, which is a transcription activator, and it packages a protein called UL41. I'm sorry, my pen does that. Which is an RNase. So RNase means an enzyme that chews up RNA. And what's really interesting is that this enzyme specifically chews up host RNA, host messenger RNA. I don't know the mechanism of how it gets to that specificity, but this is one of the ways to switch the resources from producing eukaryotic, the host cell proteins to the virus proteins. All right, here's the example of our crazy pox virus. This is the one that replicates and does transcription in the cytoplasm. This virus encodes 200 plus genes. Okay, so it encodes just about everything it needs. No virus encodes ribosomes, so all viruses use host ribosomes, but it contains its own polymerases, both DNA and RNA, and the transcription activators. It also has 3 phases: early, intermediate, and late. So it's has a temporal control of gene expression, and it packages proteins that shut down the nucleus. So there's proteins that come in here and basically shut down the host transcription and replication, and again, saving those resources for viral gene expression. Poxvirus also packages multiple virus proteins. Their proteins, or the poxvirus proteins, are packaged within the capsid, and poxvirus packages things such as RNA polymerase and cap and tail enzymes. So as soon as this DNA gets into the cell and is uncoded, you can immediately start transcription, because it has packaged in the RNA polymerase and the cap and tail enzymes, and so it makes mRNA that looks just like the host. Retroviruses, remember that they go through reverse transcription, and integrate into the nucleus. This is called a provirus. The provirus is double-stranded DNA, just part of the cell chromosome, and so gene expression is just like any other host protein using RNA polymerase, and the messenger RNAs get capped and tailed. Something a little different about retroviruses is that they make polyproteins, and we will talk about this. This is pretty common with a lot of RNA viruses. So polyproteins means that you make one long RNA and then you can cleave it into different independent proteins. I'm just going to check. Okay. Retroviruses, or HIV especially, also does something called ribosomal frame shifting, and we'll talk about this in class. Oops, ribosomal frame shift, where the ribosome is coming down, making RNA, and then it will back up, in this case, backs up one nucleotide and then continues on. And so it shifts the reading frame, and so about, I think it's on my other side, I think it's about 5% of the time it will make a GAG-Pol fusion protein. So you can see these reading frames overlap, and that GAG-Pol fusion protein, so it's GAG and Pol together, will eventually be cleaved and release the polymerase. What's interesting about this is GAG is out here on the spikes of the capsid. So you need a lot more GAG than you need the polymerase. So this is another way to regulate. So most of the time it's just making the GAG protein, and then every once in a while it does a ribosomal frame shift and makes some polymerase. So you'll see this is very common with RNA viruses as ways to control levels of protein expression. Since they're going from RNA to RNA, they don't have the same promoters and things like that that DNA viruses have in order to produce the right number of capsid proteins and lower numbers of, say, replication proteins. Hepatitis B, again, this is that weird partially double-stranded DNA. All the colors, all the fat arrows are the open reading frames. It has 4 different promoters, and it uses alternative start sites, see here, for expressing different proteins from that same gene sequence. So another strategy to have a very small genome but be able to express multiple proteins by switching the reading frames. Remember, this one's kind of cool because this guy P is for the packaging protein. And this protein actually binds the mRNA and packages it, and then remember hepatitis B uses reverse transcriptase. So I guess I shouldn't say it. It makes an RNA, uses reverse transcriptase, and then makes that partial double-stranded DNA genome inside the capsid. Okay. Because we talked about one of the issues is if you're making double-stranded RNA, the cell will respond by turning on cytokines like interferon and trying to destroy the virus-infected cell. So as it's copying some of this RNA, it does it inside the capsid to make a double-stranded genome. All right, and finally, this crazy picture is just showing you the genome of T4 phage. It's also a big 170 kilobase pair genome, 289 genes. It uses all the bacterial cell machinery, so there's no splicing, there's no cap and tail because bacteria don't use that. And if you ever see, if you're ever doing any molecular cloning and you see a T3 or a T7 promoter, they're from the T3 and the T7 phage. And these are very strong promoters. So phage have very strong promoters that highly recruit the transcription machinery to the phage genome in order to produce more phage. And kind of like a competition, it usually wins pulling in that transcription machinery as compared to the affinity of the transcription machinery for the bacterial chromosome. So it switches resources by just having a high affinity and kind of stealing away the host cell transcription machinery. All right, excuse me. So those are some of the strategies that DNA viruses use for gene expression, and in class we will focus on RNA viruses. All right, thanks.